Some proteins, they function as enzymes to speed up metabolic reaction by lowering energy barrier. So enzymes are called as biological catalyst. That's the catalyst, that's the chemical agent that speeds up a reaction without being consumed by the reaction. An enzyme is a catalytic protein to speed up biochemical reaction. Like the hydrolysis of sucrose by the enzyme sucrase is an example of an enzyme catalyzed reaction. Like this, sucrose is hydrolyzed into glucose molecule and fructose molecule. This biochemical reaction is mediated by enzyme sucrase. So without the enzyme, the process of this hydrolysis is too slow. But with the sucrase enzyme, the biochemical reaction gonna get speed up. Yeah? Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the free energy of activation, or activation energy, EA. Activation energy is often supplied in forms of thermal energy. That means that's the heat that the reactant molecule absorb from their surrounding in forms of heat, usually. As the picture showing, the reactant before the chemical reaction, they have to absorb the energy of EA, so activation energy, that much. Then the present chemical bond gonna get broken between A and B and C and D. And with the EA activation energy supply, that reactant molecule go through the biochemical reaction to become A and C and B and D product they produce. Then how enzymes speed up reactions? Enzymes catalyze the reaction by lowering the activation energy barrier. But the enzyme, they do not affect the change in free energy. Instead, they hasten reaction, they speed up reaction that would occur eventually. So, without change in free energy, the presence of enzyme, it has an effect to reduce down activation energy level. So with the enzyme, as you can see, the red line, it has reduced level of activation energy to speed up this biochemical reaction. Only one type of enzyme gonna get involved in a specific one type of biochemical reaction. So it's substrate specificity of enzyme. That means the reactant that an enzyme acts on is called the enzyme substrate. When sucrase gonna act on sucrose, then sucrose is, we call, that's the substrate. And the enzyme bind to its substrate, forming an enzyme substrate complex. The reaction catalyzed by each enzyme is very specific. So one type enzyme gonna catalyze just for one kind of biochemical reaction. So the active site is the region on enzyme where the substrate binds inside the enzyme. So I want you to remember what is active site. So that's the place for the substrate supposed to bind into. An induced fit of a substrate brings chemical groups of the active site into positions that enhance their ability to catalyze the reaction. That's the induced fit. This one is showing the substrate supposed to bind into the active site. That's why, the basic things to make binding into active site, that's the physical shape. So that's why three-dimensional shape of protein, especially for enzyme, is very important to make enzyme substrate complex. Then how an enzyme catalyzes in the enzyme's active site. In an enzymatic reaction, the substrate binds to the active site of the enzyme. The active site can lower an activation energy barrier by orienting substrate correctly, straining substrate bonds, and providing a favorable microenvironment, and covalently bonding to the substrate. This diagram showing you how the enzyme catalyzes the biochemical reaction. First one, substrate enter into the active site. Then we can see the enzyme substrate complex they produce after binding. And those substrate, like the sucrose, substrate are converted to product. This product from sucrose, it gonna get broken into glucose and fructose by hydrolysis. Then we can see the products released out from the enzyme. Then enzyme's active site gonna become empty again. So it's gonna get available for new substrate, like the new sucrose molecule can bind into the empty active site of the enzyme again. and the effects of local conditions on enzyme activity. Enzymes activity can be affected by general environmental factors such as temperature and pH, and chemicals that specifically influence the enzyme. And for the effects of temperature and pH, each enzyme has an optimal temperature in which it can function. Each enzyme has an optimal pH in which it can function. And optimal conditions favor the most active shape for the enzyme molecule. That's why. Some protein, they need some additional thing for their function. We call that's the cofactor. So cofactors are non-protein enzyme helpers. So cofactors may be inorganic, such as a metal in ionic form, like Fe++ or Cu++, something like that, or organic. An organic cofactor is called a coenzyme. Coenzymes include vitamins. Some chemicals, they act as enzyme inhibitors. So we call that the inhibition, enzyme inhibition. So this inhibition is sometimes bad because when you take some poison they act as a enzyme inhibitors. So it can inhibit some enzymatic biochemical reaction. It can stop biochemical reaction, so that's too bad. However, sometimes this enzyme inhibition is going on purpose because for the control the biochemical reaction, sometimes we need to stop some biochemical reaction. That's good for the body. So that's why enzyme inhibition, it can be by the accident or it's too bad, like poison, or enzyme inhibition, it can be good as a process for the controlling biochemical reaction, controlling metabolism inside the living body. So not all the time bad. So enzyme inhibitor, we can see sometimes competitive inhibitors bind to the active site of an enzyme. So those inhibitors, they bind into the active site, then proper substrate cannot bind into the active site, competing with substrate. The other one is non-competitive inhibitors bind to another part of an enzyme, not into the active site directly, but they can bind some other part than active site, causing the enzyme to change shape and making the active site less effective, so it can stop functioning as an enzyme. So examples of inhibitor include toxin, poisons, pesticides, and antibiotics. The picture showing the comparison among the normal binding for the normal biochemical reaction, that's the A, and B, that's the competitive inhibition, and C, that's the non-competitive inhibition. First time, for the normal biochemical reaction, the normal substrate gonna get bind into the normal active site of an enzyme. That's for the start of a biochemical reaction. And second one, that's the competitive inhibitor. That's not the substrate, but their shape is very similar to the normal substrate, but that's not substrate, but inhibitor. So they compete each other for the same active site. So when those competitive inhibitor bind into the active site first, then normal substrate, they cannot bind into the active site. So no more biochemical reaction. So biochemical reaction gonna get stopped because of the competitive inhibitor. And the third one is showing the non-competitive inhibition. That inhibitor does not bind into the active site. Instead, it's gonna bind into some other site than active site. But those binding, it can cause the overall shape of the enzyme, so it can deform active site shape. Then what? Normal substrate cannot bind into the active site anymore. So that's why the biochemical reaction with the enzyme gonna get stopped. So that's the inhibition, non-competitive inhibition. So as we just discussed, the inhibition of an enzymatic activity or stimulation of an enzymatic activity, it helps to control the metabolism. So regulation of enzyme activity helps control metabolism. So chemical chaos would result if a cell's metabolic pathway were not tightly regulated. It can cause a lot of trouble. A cell does this by switching on or off the genes that encode specific enzymes, or by regulating the activity of enzymes. So a type of regulation of enzyme, we can see allosteric regulation of enzyme. Allosteric regulation may either inhibit or stimulate an enzyme's activity. So sometimes they're gonna inhibit biochemical reaction, or sometimes they stimulate the enzyme activity. So allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. Most allosterically regulated enzymes are made from polypeptide subunits. So those enzyme is composed of many subunits. Each enzyme has active form and inactive form. They have two forms. One active form, the other one is inactive form. So of course the active form, they stimulate the biochemical reaction, and with the inactive form, those biochemical reaction gonna get stopped. Yeah? That's the active form and inactive form. The binding of an activator stabilizes the active form of the enzyme. Then what? They stimulate those biochemical reaction like this. And the binding of an inhibitor stabilizes the inactive form of the enzyme to stop, to inhibit the biochemical reaction. Those diagram shows you that allosteric activation or allosteric inhibition and cooperativity. So two types we can see. First one, A, in allosteric activation and inhibition, that what? When you look at the enzyme, that allosteric enzyme composed of 1, 2, 3, 4 subunits. And that one, first one, we can see that's the active form, yeah? Active form. So with the active form, activator, when the chemical activator, some chemical, they act as an activator. So when those activator bind into the some part of the enzyme, it stabilizes the active form. So with those active form, those biochemical reaction, it can going on and on, yeah. So it can stimulate the biochemical reaction. However, those allosteric enzyme, when inhibitor bind into the site, then what? Then those inhibitor, it can stabilize the inactive form. So with the inactive form, the substrate cannot bind into, so no more biochemical reaction. Yeah? So biochemical reaction gonna get stopped. Yeah? And on the right hand side one, it's showing you cooperativity. So when substrate bind into the active site, that binding itself, it can stabilize the active form. Yeah? So the presence of substrate, it can accelerate the biochemical reaction. Yeah? So we call that's the cooperativity. Cooperativity is a form of allosteric regulation that can amplify enzyme activity, because one binding of a substrate, it can secure the active form of allosteric enzyme. So one substrate molecule primes an enzyme to act on additional substrate molecules more readily. So cooperativity is allosteric because binding by a substrate to one active site affects catalysis in a different active site. Feedback inhibition. Sometimes a cell, they produced too much product by a enzymatic activity, then they need to stop. So in that case, final product, it can act as an inhibitor for some enzyme to stop the biochemical reaction. We call feedback inhibition. In feedback inhibition, the end product of a metabolic pathway shuts down the pathway. So feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. This diagram shows one example for feedback inhibition. So isoleucine used up by cell. Then the enzyme gonna get activated. So first step, threonine gonna get bind into the active site of this enzyme to produce intermediate A, and with another enzyme it turns into intermediate B and C and D, and through the intermediate D with an enzyme the end product gonna get produced as isoleucine. However, when they produce too much isoleucine, they act as an allosteric inhibitor. So they bind into the allosteric site of that first enzyme to make stop. Then active site no longer available, then the pathway to produce isoleucine gonna get stopped, get halted. That's an example for feedback inhibition. The last class of biological molecule, that's the nucleic acid, DNA and RNA.